biochemistry, the last part where we're going to go over the different levels of structure. Yeah!
I love chemistry. Yay, yay, yay. Ha ha ha. Ha ha ha.
Alright, let's move on to quaternary structure, and I'm going to come back to myoglobin, which is the primary example that I'll go over. Myoglobin is going to be our tertiary protein, globular tertiary protein example. Myoglobin, of course, is in the muscle cells, and it's going to be holding on to oxygen for the muscle. Whereas hemoglobin, which is a quaternary globular protein that is in the blood, actually in the erythrocytes, the red blood cells, that is going to be holding on to the oxygen from the lungs throughout the system, letting it go at the tissues. In the muscle cells, we're going to have the myoglobin kind of holding a temporary storage of oxygen. We have both of these globular proteins working together. One of them is tertiary, one of them is a quaternary. So structure simply is going to be a complex of two or more tertiary units. So we're going to have potentially just 2 units. We can have 3 units, we can have 4, maybe more. In our example here, hemoglobin is going to be 2 alpha units, 2 beta units. So it's ultimately 4 separate polypeptides complexed together into a functional protein. So for a quick review, we had primary structure, which was a linear sequence of amino acids. The structure, which is the sequence of amino acids, is going to equal the ultimate function. The function is going to be governed by the folding of the protein that will be assisted by chaperone proteins right after they're translated from a ribosome. Once we have a folded protein, it will either be in the tertiary 3D structure, or depending upon the kind of protein it is, it could be in a quaternary complex. Whatever it is, the folded protein is in its native conformation, which is how it is intended to be folded by its linear sequence. And this is going to be the most energetically favorable folding pathway.
Hey, Dr. Bond. Can you please teach me about myoglobin and hemoglobin? I want to know the difference.
Well, I can answer that. Okay, now let's talk about what's very important, because I get really excited when we get to something that's close to physiology, because you know what? I'm a physiologist. Okay, so anyway, myoglobin, as we discussed, is a tertiary 1 unit protein. It's globular. And then we have hemoglobin, which is a quaternary structure, which means that it has more than 2 subunits, and hemoglobin has 4. We have 2 alpha chains and 2 beta chains in a hemoglobin, and only 1 lonely chain in a myoglobin. But the myoglobin chain and then the alpha chain and the beta chain of the hemoglobin, they're homologous in a way that their amino acid residues are pretty close to the same, you know, in the sequence. Because they're both going to be carrying a prosthetic group, and a prosthetic group is any molecule that's bound to a protein that aids its function is a prosthetic group. So it's a non-amino acid that's helping the protein function. And so the prosthetic group in the case of both the myoglobin and the hemoglobin is a heme unit. The heme unit is going to be where the oxygen will bind to. Remember that both the function of the myoglobin and the hemoglobin is to carry oxygen, so it will do so at the heme group. Now, a myoglobin has 1 heme group, so therefore it can carry 1 oxygen. The hemoglobin has 4 heme groups. Even though it has 2 alpha chains and 2 beta chains, they both have a heme group, so each hemoglobin molecule has 4 heme groups and can carry 4 oxygen molecules. Now, just to show you the relative size, the alpha chains, which are the 2 identical chains, subunits in a hemoglobin, they're 141 amino acid residues long. Beta chains are 146 amino acid residues long. And then the myoglobin single polypeptide is 153 amino acid residues long. So they're about the same size polypeptide chains in both the myoglobin and then both of the types of subunits of a hemoglobin. Okay, so let's look how both the myoglobin and the hemoglobin bind to an oxygen molecule. Let's look at hemoglobin because it's going to be the same in myoglobin. We have a deoxyhemoglobin, which means it doesn't have oxygen, and then we have an oxyhemoglobin, which means it's carrying an oxygen. Now in the deoxyhemoglobin, the heme group is actually planar, so it's going to be on a plane, it's flat. but it's actually slightly kind of puckered a little bit in the deoxyhemoglobin. Now the iron is in the heme molecule, and this is going to be what actually binds to the oxygen. And when you put an oxygen, when you bind an oxygen to the heme group, what it does is it actually physically lifts the iron molecule and it makes it more flat. So it slightly changes the shape of the heme group. And so when we bind this oxygen to the iron of the heme group, then we become oxygenated and change the shape ever so slightly. Now in the case of a hemoglobin, it exhibits positive cooperativity, which we'll go over with enzymes. But what that means is because it has 4 different subunits, when one of the subunits binds with its favored molecule, whatever it is, in the case of an enzyme, it's going to be whatever like a lock and key fits into its active site. Here we're talking about a heme group that has an iron that wants to bind with an oxygen. When it binds one of its subunits, then it's going to encourage or enhance the binding of its other subunits to bind to the same. So that's positive cooperativity. So once one oxygen binds, then now all of a sudden the other heme unit really want to bind to an oxygen. So again, positive cooperativity. How does that happen? Like I was explaining before, on the heme group that has an iron atom, when it binds to the oxygen, if you look at the structure, in the deoxyhemoglobin versus the oxyhemoglobin, we're going to lose the puckering and becomes a little bit more like a fraction of an angstrom. It becomes a little more planar, more flat, and what this does is it's going to pull the fraction of an angstrom on, say, the histidine that the iron is attached to, which is thereby attached to another amino acid and so forth in the chain. So if you move one, you're going to, like dominoes, you're going to be moving it until you ultimately change slightly the entire shape of the subunit. And what that does is that's going to slightly affect how it interacts with another subunit, and it's going to affect its shape, thereby allowing it to bind to oxygen more freely, and so forth. So it's encouraging the other subunits in the molecule to bind to oxygen once one of the heme units has one of its iron groups bound to an oxygen. So again, that's positive cooperativity. And we'll see this activity in some enzymes. Let's look at a comparison of how myoglobin, which is going to be in a hyperbolic plot, and hemoglobin, which is going to be in a sigmoidal plot. Let's look at a graph of how they both bind to hemoglobin. So myoglobin is going to be 50% saturated with hemoglobin. Remember, it has 1 heme unit per molecule at 1 torr. And if you remember that 1 torr is equal, like 1 atm is equal to 760 torr. So it's just a measure of pressure, how much pressure oxygen is exerting in the atmosphere. Hemoglobin, on the other hand, does not reach 50% saturation until we get to 26 torr. So the lung pressure of oxygen is going to be about 100 torr. At that point, both hemoglobin, well, myoglobin's not in the lungs, but if it were in the lungs, it would be 100% saturated at 100 torr. And then at an actively respiring tissue, the pressure of oxygen is about 20 torr. So hemoglobin is going to be less than 50% saturated at 20 torr, whereas myoglobin is still going to be up there closer to 100%. Why? Because myoglobin is a storage molecule of oxygen, and hemoglobin is the transport molecule of oxygen. So we want hemoglobin to let go of the oxygen at the actively respiring tissues. We want to use up our hemoglobin resources first and then use myoglobin as like the backup. So myoglobin, if you look at this graph, at 1 torr of partial pressure, it's 50%. So bam! It doesn't take that much pressure of oxygen for it to be pretty saturated. Again, it's a storage molecule. Myoglobin is only going to give up oxygen when the partial pressure of oxygen is very low. So once the hemoglobin storage has been exhausted, then myoglobin can act as a backup. So when you work out, hemoglobin probably will not keep up with you. So that's why you have myoglobin in your active tissue. When we look at this graph and we see at a low concentration of oxygen, not a lot of it is going to be bound to either myoglobin or hemoglobin, but as you go up, you can see that myoglobin will retain the oxygen much better than hemoglobin does. So when oxygen concentration is low, such as with the actively respiring tissues, it's going to be at about 20 torr. And we've already discussed at 26 torr, the hemoglobin is 50% saturated. So at the tissues, we're going to have negative cooperativity to where when oxygen travels from a high to low concentration and is released from the hemoglobin, then we're going to have the cooperativity again where we're going to be changing from the R state to the T state, and we'll go more over that when we talk about enzymes. Okay, so at the lungs, again, we're going to be at a partial pressure of 100 torr, and the hemoglobin will be 100% saturated. Then it's going to travel around the body, and at the capillary beds of actively respiring tissues, it's going to be at about 20 torr. And if we look at our graph, here we can see that hemoglobin is going to be about 30% saturated, and that's because it's releasing the oxygen at the tissues, so it's a perfect delivery system. Alright, the deoxyhemoglobin versus the oxyhemoglobin, these are in two conformational shape. We have what is known as the relaxed shape, or the R shape, and then we have the T shape, which is the tight shape or taut shape. If you think of it, if you're very tight, then you're kind of stuffy and you don't want anything. That's kind of like a hemoglobin that doesn't want any oxygen. That would be at the tight shape. At the R shape, you're relaxed and you want oxygen. So once we bind one of the oxygen molecules to a heme group of hemoglobin, then we're going to conform to the R shape. It's going to be relaxed. The positive cooperativity is going to draw in the other three oxygen. Very high affinity for the binding of oxygen in the R state. We have two other molecules that can bind to hemoglobin and affect the oxygen binding or oxygen affinity, and that is hydrogen ions and carbon dioxide. And as you know from metabolism, from what you studied thus far, that CO2 is going to be a byproduct of metabolism. And let's talk about the hydrogen. So actively respiring tissue is going to be releasing hydrogen, therefore it's going to be lowering the pH. Therefore, it's going to be decreasing the affinity of hemoglobin to oxygen. An increased amount of hydrogen ion is going to protonate key amino acids and thereby decrease the affinity of the hemoglobin for oxygen. So that makes sense. So you have an actively respiring tissue. It is going to become more acidic because we're releasing hydrogen ions into the local environment. The hydrogen ion is going to bind to the hemoglobin and reduce the affinity for oxygen and therefore encourage it to let go of the oxygen at the tissues that are actively respiring because of the low pH. Myoglobin, on the other hand, is not affected by this. And this effect that hydrogen ions have on hemoglobin is called the Bohr Effect. And if we look at a graph, we can see as the pH is lowered, so is the affinity of hemoglobin for oxygen. So again, you have an actively metabolizing tissue that's going to be releasing hydrogens, lowering the pH. By doing that, we're going to reduce the affinity of oxygen that the heme group has for oxygen. It's going to release the oxygen, and then it will travel via simple diffusion from high to low concentration towards the tissue that needs it. So the acid-base properties of hemoglobin will affect the oxygen binding properties. The oxyhemoglobin is going to be a stronger acid. It's going to have a lower pKa than the deoxy version of the hemoglobin. Thus, the change in the hemoglobin structure also can act as a buffer in the blood. Now let's talk about something that we'll talk about when we talk about glycolysis later. It's a molecule called 2,3-BPG or bis-phosphoglycerate, and this is going to be released by actively respiring tissue. So not only will actively respiring tissue release hydrogen ions, it's going to be releasing 2,3-BPG. And if you look at the structures of hemoglobin from the top down, you see that oxyhemoglobin has like a donut shape, and this 2,3-BPG via electrostatic attractions, the positive and negative attractions, will fit into this donut hole, and it will basically lock it into the T shape. So it's going to favor the T shape over the R shape. And remember, a T shape doesn't want oxygen, so it's going to reduce the affinity for oxygen. So T3-BPG, which is released by actively respiring cells, muscle cells that are actively undergoing metabolism, cellular respiration, we release it into the local environment. The deoxyhemoglobin is going to pick it up and it's going to be favored in the T shape. If this continued, if it stayed locked in there and it got to the lungs, what would happen? Well, the hemoglobin wouldn't want to pick up oxygen and that would be a problem. So this is just electrostatic attraction, so it goes in and out, in and out, and in and out. And it just depends if you have a high concentration of T3-BPG, then chances are you're going to just overall reduce the affinity for oxygen. One exception to this, I'm sure you've seen people that, are not very active athletically, and if they try to be, they can't breathe very well, and it's self-induced. Smokers, you have more T3-BPG in your deoxyhemoglobin that is locking it into a T state. So you're doing this to yourself, so stop smoking. Okay, if you want to be able to breathe better, stop smoking and you're going to reduce your T3-BPG and you'll feel better. Okay, so for those of us that don't smoke and that just have the normal hemoglobin actions with T3-BPG, what happens is that as the hemoglobin gets back into the circulatory system, it's going to release the T3-BPG and another cell will take it up and metabolize it. Okay, so it doesn't stick with it all the way to the lungs and block it from picking up oxygen. Okay, let's talk about little babies, little fetuses that are growing inside the mommy's uterus. How does their hemoglobin compete with mom's? Because you need some kind of draw if you're a fetus. Mom has this extensive circulatory system, and you can say we'll have first dibs on any oxygen that she breathes in, and as you know that the placenta is going to be an interface of mom's circulatory system with the fetal circulatory system, but they're going to, share. So the oxygen travels via this placental interface into a fetus. Now, how does the fetus grab the oxygen from mom? So what we need to do, if you're a fetus, is have a stronger affinity for oxygen than mom does, and the fetus does this. It will have a slightly altered structure of its hemoglobin. Instead of having 2 alpha and 2 beta chains, it has 2 alpha and 2 gamma chains. These 2 gamma chains have a higher affinity for oxygen than mom's beta chains do. So it's going to be pulling the oxygen away from mom. Now, does that affect the fetus from being able to let go? No, not really, because we have something else. Basically, the gamma chain is different from the beta chain because we've substituted a serine for a histidine. It's actually histidine 143 that is substituted with a serine, so it's just a slight change in structure, but it's enough of a chain to where we don't have this donut shape anymore, so we can't have the T3-BPG blocking it. So the fetal hemoglobin is going to be in a constant R state at all times. So we're always going to be picking up oxygen, and because the fetus doesn't really have a lot of activity other than maybe kicking the crap out of mom every now and then randomly, it's not a problem for a release of oxygen. Okay, one more thing that we want to touch on is the large amount of CO2 produced by metabolism. Again, we'll talk about that later with Krebs cycle, but that's going to be forming carbonic acid. And we did talk about buffers before with water chemistry. Carbonic acid is H2CO3. The pKa of H2CO3 is 6.35. The normal pH of blood is going to be 7.4. So as a result, 90% of the dissolved CO2 is going to be present as bicarbonate ion, which is HCO3 negative, and it's going to be releasing hydrogen. Thus, we have an in vivo buffer system. We have hemoglobin acting as a buffer, as well as this bicarbonate system, the H2CO3 and HCO3 negative plus hydrogen ion. Okay, the presence of a large amount of hydrogen ion is going to favor the deoxy form of hemoglobin. Thus, it's going to lower the affinity of hemoglobin for oxygen. The bicarbonate is going to be transported to the lungs via the bloodstream, where it is going to combine with the hydrogen ions that are going to be released by hemoglobin when it binds to the oxygen and produce the H2CO3. In turn, the H2CO3 is going to liberate a CO2 that you're going to thereby exhale at the lungs. Hemoglobin is also going to directly transport some of the CO2 that is liberated at the cells of active of actively respiring cells.
This is Brenda, the not-so-good witch, signing off for today. See you next time on Dr. Bond's Science Theater.